Welcome people. What you are seeing on your screen is a WBC, a white blood cell, which is a typical eukaryotic cell. So this is a schematic diagram taken from an actual image. So what is an eukaryotic cell? As most of you might have known, it is something, a cell kind which will have a nucleus which will be covering the DNA or the genetic material. Now a prokaryotic cell does not have a nucleus, and in this image you can clearly see the central dark part is the nucleus. Now covering it we have cytoplasm, which carries all the proteins and RNA molecules getting translated and all the other molecules. Now covering the cell is the plasma membrane. A plasma membrane mostly consists of lipid and proteins. Being part of all living cells, the plasma membrane has some important roles to play, apart from just being a cover or envelope to the cell. Now we have to look at the composition of the plasma membrane. So it consists of mainly lipids, and along with lipids we have different proteins, and in some fraction we also have polysaccharides or carbohydrates. Usually these carbohydrates are present as glycoproteins, where they are attached to some protein molecule, or glycolipids, where they are bound with some lipid molecules. Let us look at the structure of the lipid. It has a polar head, which is usually a glycerol molecule along with a phosphate moiety, and then it has two fatty acid chains. Well, the fatty acid chains are non-polar. So the lipid molecule is usually called amphipathic molecule because it has a polar head. The polarity comes from the negative charge of the phosphate group, while it is a hydrophobic or non-polar tail, which comes from the fatty acid, which are organic in nature. Let us look into the properties that the lipid bilayer gets from the lipid components. First, if we have long or saturated fatty acids, then the structure will be less flexible, as these fatty acids cannot move in a lateral direction much. So this will make it less fluid. Whereas if we have short fatty acid chains or unsaturated fatty acid chains, it will keep the lipid molecules far apart, and then will have more flexibility. As shown here, these lipid molecules are far apart from the ones depicted above because of their shorter and unsaturated fatty acid chain. The double bond can be seen on the chain. This makes it more fluid, which is in different terms more flexible. Let us look at some of the models that were proposed on the structure of the lipid plasma membrane. The most simplest and the first model that was proposed was of a lipid bilayer, which consists only of two layers of lipid molecules. This is a typical structure that a liposome will have, where we have one layer of lipid on the top and another layer of lipid on the bottom facing opposite, so that the hydrophobic side will stay inside and the hydrophilic side will be exposed to the outer medium, which can be water. And this will keep the hydrophobic core away from polar molecules like water. However, this model fails to explain the presence of different proteins. in the plasma membrane. So then came the sandwich model. The sandwich model suggests that the lipid molecules are sandwiched between layer of proteins. Now the proteins are usually charged, so they can interact with the polar head of the lipid molecules, and then the hydrophobic part will stay inside, safe from the polar molecules outside. Here we have the proteins depicted in yellow, and it assumes that all the membrane proteins will stay on the surface of this cell and sandwich the lipid bilayer in between. However, not all the proteins are present outside the cell surface, and it has been observed that there are some proteins present spanning the whole lipid bilayer from outside to inside of the cell. There came another model called the fluid mosaic model or quasi-static model. Now this model assumes proteins as icebergs inside a sea of lipid molecules. So in diagram we can show that there will be a lipid bilayer model, but in between this bilayer there will be proteins incorporated, and the proteins can span the whole bilayer as depicted in that green mark, or they can be embedded into the layer partially and exposed to the extracellular or intracellular side partially.